Okay, welcome back. This is lecture 3, part 3. This is the last part in this lecture. Okay, we talked about our first and second lectures. We talked about the drugs that interact with nucleic acids, DNA and RNA. In the first and the second parts from this lecture, we talked about anti-metabolites or anti-metabolites. Now let's talk about hormone-based therapies. Okay, so basically using hormones to treat cancer. Okay, hormone-based therapy is used for cancers which are hormone dependent. What does that mean? That means there are some kinds of cancers or neoplasms that depend or need hormone to grow. Okay, so either by giving hormones which have the opposite effect, if we know which hormone the cancer needs to grow, we can either give another hormone which have the opposite effect, or by administering a hormone that antagonizes the effect of the hormone that the cancer needs. Okay, oh sorry, yeah, that's the first one, giving a hormone that antagonizes the effect of the hormone that the cancer needs, or giving hormone antagonists that antagonize the hormone that the cancer needs. Or we will see that sometimes we will even give some kinds of hormone agonists, but we will talk about those later. The first one are the glucocorticoids, estrogens, progestins, and androgens. A lot of hormones that are used in cancer treatments. Example on those like the glucocorticoids, prednisone and prednisolone. And as you know, prednisolone is a derivative from prednisone, or you could say like prednisone is a product for prednisolone. Okay. Also, estrogens have been used in cancer treatments. Why? Because estrogens decrease the levels of luteinizing hormone, and if you decrease the luteinizing hormone, this will lead to decrease testosterone. Okay? And some cancers need testosterone to grow. Examples on the estrogens are the ethinyl estradiol and diethylstilbestrol. The ethinyl estradiol is a derivative of estradiol, and the diethylstilbestrol is non-steroidal estrogen. Okay, it's just like you have non-steroidal anti-inflammatory drugs, you have non-steroidal estrogen. Okay, the phosphosterol, which is a product for the diethylstilbestrol, also used as an estrogen in cancer treatment. These are the structures for the medication that we just mentioned. You see here the prednisolone and the prednisone and their structure, and as we said, prednisone is a product for prednisolone. So prednisone has carbonyl group at carbon number 11, you just get reduced to a hydroxyl group, alcohol. This is the structure of estradiol. In case of estradiol, this one is hydrogen. For the ethinyl estradiol, and as the name implies, has the ethinyl group, the C triple bond CH here instead of the R group. This is the diethylstilbestrol, and as you can see, all of these three contain the steroid nucleus. We, we call these 4 rings steroid nucleus. Okay, this one doesn't have the steroid nucleus, so it is a non-steroidal estrogen, the diethylstilbestrol. The phosphostrol, which is the phosphate product of the diethylstilbestrol. Here are the R group. Okay, other examples on hormones that have been used for cancer treatments are the progestins and the androgens. Okay, the progestins used as an anti-cancer agents include the hydroxy, progesterone acetate, and the megestrol acetate. And I'm not gonna go here in deep about what the hormones do and how they exert their effects, but I'm not gonna go in deep in that. I think you already studied the hormones. Most of them exert their effect by negative feedback. Not all of them, but most of them. Also, androgens decrease the levels of luteinizing hormone. If you increase androgens or you give androgens, you will decrease the luteinizing hormone, which will eventually decrease estrogen synthesis, because you need luteinizing hormone to induce estrogen synthesis. Examples on androgens used in cancer treatment: floxy, misterone, and the testosterone propionate, which is a prodrug also, just like the others. It's a prodrug for dihydrotestosterone. These are the structures for the hydroxyprogesterone acetate, the megestrol acetate, the floxymisterone, and the testosterone propionate. You see that all of them has the steroid nucleus, okay, except few changes, just a few differences in the substituents, okay, and this is the dihydrotestosterone. Again, if you have any question, don't hesitate to ask. Ask now before it's the exam time, okay? All right. Other examples on medications used for cancer treatment that are related to hormones are the luteinizing hormone releasing hormone agonists. Okay, so we have the luteinizing hormone and we have the luteinizing hormone releasing hormone, LHRH, or sometimes they call it the gonadotropin releasing hormone. Okay, they found that long-term exposure to LHRH will desensitize the receptor. Okay, that will lead to drop in luteinizing hormone levels. Okay, so they thought about giving the luteinizing hormone releasing hormone, but the luteinizing hormone releasing hormone, as you can see from the figure, it's a decapeptide, and you know what deca means. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 amino acids. Decapeptide, 10 amino acids peptide. Okay, so if you give a peptide, it's gonna be what? It's gonna be degraded inside the body because of the effect of esterases and the peptidases. Okay, so they made analogs of the luteinizing hormone releasing hormone, and those analogs have an agonist effect, so they have the same effect as the LHRH. Example of those analogs: the leuprolide and the gosarline. Both are decapeptide analogs of LHRH. They just modified the amino acid at position number 6. In the LHRH, the amino acid is glycine. They used the unnatural amino acid, They use the D amino acids. As you know, the natural amino acids are the L amino acid, the levo. They used the dextro amino acid instead of the levo amino acids. They found that this has better stability, and just change the amino acid at this position will make this decapeptide a non-substrate for the peptide. Okay. Also, changing the amino acid glycine at the position number 10 with suitable group like those two will make the analog, will increase the affinity of the analog to the receptor. Okay. Okay, let's move on. The anti-estrogen. We said that we give estrogens earlier, right? Now we give anti-estrogen because there are some cancers that are estrogen dependent. Example of those is the breast cancer. Example on the anti-estrogens are the tamoxifen and paroxetine. You see the structure here. For tamoxifen, R group is methyl and the X group is hydrogen. And this is the structure of paroxetine. Okay. Newer analogs, newer anti-estrogens are the chloromethine and the fulvestrant, which are more recent anti-estrogens. This is the structure for the fulvestrant and is just approved in 2002, which is relatively new if it's as compared to the tamoxifen. If you can have a look at the structure, you see that this corresponds, or this is an analog for the steroid nucleus. Okay, and it has a branch here or a side arm. This side arm or this branch is important for activity. This is the side arm for the tamoxifen. This one is for the raloxifene. So why it's important for activity? Well, before we go there and before we see why it is important for activity, let us have a look at how the estrogen works on the estrogen receptor. Okay, let's take example as the estradiol, as you can see here from this little cartoon. When the estradiol attached to the binding site at the estrogen receptor, this will induce change in the receptor. Okay, that will flip down the H12 part of the receptor. Okay, it will flip as a lid, just like a lid or a cap on the estradiol or the estrogen binding site. This will expose the AF2 region. Okay, then dimerization and exposure, as we said, of the AF2 regions will happen. After that, the co-activator will come and interact with the dimer at the AF region, AF2 regions. This whole complex is called the nuclear transcription factor. Okay? This nuclear transcription factor will interact with DNA and lead to transcription. Transcription will form RNA. RNA will make proteins, and proteins will do their work. So what the anti-estrogens do? The anti-estrogens, as you saw from the structure, this is resemble the steroidal nucleus, and this is the side arm. So what will happen? The steroidal nucleus will interact with the binding site, just like the estrogen itself. Okay, so this will interact with the binding site. The hydroxyl group will form a hydrogen bonding with histidine 524, and the other hydroxyl group will form two hydrogen bonding, one with glutamate 353 and the other one with arginine 394. Okay, but we said that the side chain or the side arm is important. Why? Because the amino group will be protonated, okay, and form a hydrogen bond to aspartate 351. Okay, by forming that bond, the side chain will prevent the receptor helix, as we said, the H12, folding over as a lip. So this side chain will protrude out, and this will prevent the helix H12 from folding over as a lid. Okay, this will result in the AF2 binding region that is not exposed, so the co-activator cannot bind, and that will prevent the formation of the nuclear transcription factor and eventually prevent DNA transcription. I hope you got it. All right. We talked about the anti-estrogens. Let us talk about the anti-androgens. Okay. Example on those are the flutamide and cyproterone acetate. This is the structure for flutamide, and this is the structure for the cyproterone acetate. They work by blocking the action of androgens at their receptor, just like the anti-estrogen. The abiraterone, the structure of the abiraterone, is a potent and selective inhibitor of a metabolic enzyme. So this one, although it has the steroid nucleus, just like these, but it will not act on the androgen receptor. Okay, this one is an inhibitor, a selective inhibitor, potent selective inhibitor of an enzyme that is called 17-alpha hydroxylase 1720 lyase. They found that by inhibiting this enzyme, you can inhibit the androgens synthesis, just like testosterone. And the prodrug, the abiraterone acetate, has the methyl as the acetate group at the R position, this one, instead of hydrogen in the abiraterone. One important thing to mention here that this enzyme has an iron heme in it. It has a heme part that has an iron. The pyridine ring is essential to interact with the iron and stop the effect of the enzyme, stop the action of this enzyme. Okay. Last but not least are the aromatase inhibitors. The aromatase enzyme catalyzes the last stage in the biosynthesis of estrogens from androgens, okay, where an aromatic ring is formed, and hence the name aromatase. As I told you, most of the enzyme names are straightforward. They just name them directly. Either they name them according to their substrate or according to their product, or sometimes, just like this one, it depends on the action of the enzyme. So what the enzyme do, the enzyme will make an aromatic ring. Okay, so they call it aromatase. This enzyme consists of two parts. It's a complex of the cytochrome P450 enzyme containing heme, which means contains iron, and a reductase enzyme using NADPH as a cofactor. Eventually, you are going to need a reductase to make the aromatic ring. The cytochrome part, or the cytochrome enzyme, bind the steroid, bind the androgen substrate, and catalyze the oxidation. Okay, it is considered as an important target for anti-estrogenic drugs because it catalyze the last stage of the biosynthesis of the estrogen. Okay? There are two types of inhibitors for the aromatase, either reversible competitive inhibitors or irreversible suicide inhibitors. And I think you would know that irreversible, it means that it's going to form covalent bond with the enzyme. Okay. This figure shows you, or this scheme shows you the action of the aromatase enzyme. Okay, so the aromatase will take an androgen molecule like testosterone or androstenedione, this molecule, and it will use NADH or NADPH, three molecules of the cofactor, and three oxygen molecules to make the estrogen, like either estrone from androstenedione or estradiol from testosterone. This is the action of the enzyme. And in the process, this will release formic acid. Okay, all right. We said that we have two types of inhibitors. So the first one are the aminoglutathione, which are an early examples of a reversible competitive inhibitor. Okay? They inhibit the enzyme reversibly, but it has disadvantage. It binds to various cytochrome P450 enzymes because, as we said, the aromatase has the cytochrome P450 enzyme and a reductase enzyme. It's a complex enzyme. So the aminoglutathione bind to various cytochrome P450 enzymes. That's why it inhibits a range of steroid hydroxylations. So it has a lot of side effects. But newer agents based on the aminoglutathione as a lead compound, newer agents have been designed. Examples on those are anastrozole and letrozole, which are more selective inhibitors and used to treat breast cancer. Why we use them to treat breast cancer? Because breast cancer is what it depends on what hormone? Estrogens. And aromatase make estrogens from androgens. So if we stop aromatase, we will stop the synthesis of estrogen and we will stop the growing of the breast cancer. It is used as an alternative therapy to breast cancer that are resistant to tamoxifen. These are the structures for the aminoglutathione, anastrozole, and letrozole. The anilino group, or the anilino nitrogen, along with the N4 of the triazole, of the anastrozole, and the letrozole, all of those are important because they interact with iron at the heme group, in the cytochrome P450. I think now you have an idea what groups we need if we want something to interact with iron. All right. Other inhibitors of aromatase, as we said, we have the reversible and we have the irreversible. The formistine act as a suicide substitute. It has the steroid nucleus. Okay, so it acts as a suicide substrate for the aromatase enzyme. It permanently inactivates the enzyme and it is more selective than the aminoglutathione. Okay. Thank you very much for your listening, and this is the last or the end of the last part on this lecture. I hope we will meet you after everything ended, and I hope we will meet you in the college for other lectures. Goodbye.